This pharmacology lecture was recorded in spring of 2015 on the topic indicated at the top of the slide. Please note that this lecture is quote-unquote as is, meaning that at times the audio quality may not be sufficient to completely hear everything as it was initially presented. There is no substitute for regularly attending class.
Now, the next type of receptor I'd like to talk about, we've got about 10 minutes left, are the ligand-gated channels. Now, the ligand-gated channels, I don't know where you're going to write it, but I would just say fast responding. Fast responding. And so there are certain ligands which use these kinds of channels, especially neurotransmitters, acetylcholine, GABA, certain excitatory amino acids. And these channels allow for rapid information transfer. So let's imagine for the moment that you're a sodium ion, and you're looking down on a cell, and you're seeing the polar head groups, and it almost looks great, like you could just go on in, but you can't because you see those hydrophobic tails. And you'd love to go into this thing, but this thing's closed. This is the nicotinic receptor. So the nicotinic receptor is a humongous protein in the membrane. It's made up of 5 subunits, 2 alphas, a beta, a gamma, and a delta. Each subunit is at least 43,000 times the size of a hydrogen atom. So this thing is huge, right? And somewhere you should write down, because I don't have my mouse today, non-selective cationic channel. Non-selective cationic channel. So it will allow the passage of sodium into cells. Some studies have shown it will allow the passage of calcium into cells. Some studies have shown it will allow the passage of potassium out of cells. So that's why we say non-selective cationic. The major ion that goes through it is sodium. Okay, so in the panel below, we have the same situation, but acetylcholine has been bound. And in fact, 2 molecules of acetylcholine bind the channel on each alpha subunit, and the channel opens, and sodium can now enter into the cell. And so we know a lot about the nicotinic receptor. It's been around for thousands of years. But in research terms, scientists studied it at length in the 1950s. They harvested it from a sea creature called an electric eel. And so the electric eel it's known by a scientific name as torpedo. And so this eel is able to generate electrical current, I guess as a way of defending itself from a predator. And it uses these nicotinic channels to do it. So we originally isolated nicotinic receptors out of torpedo, and interestingly enough, these receptors are conserved all the way up to man. Right? So they're all similar as you go up through evolution. Again, very bulky subunits, acetylcholine binds, conformational change, channel opens, cation moves. Now, the interesting thing about this is that you can measure cellular response on the millisecond scale. So much different than what we saw for example, steroids, or for example, thyroid hormone, or for example, vitamin D. Right? And this is interesting because this allows rapid transfer of information. So for whatever reason it was designed this way, because it's designed this way, you can hear my voice, and very rapidly, these channels are opening up in your brain, they're resetting, they're opening, they're resetting, they're opening, they're resetting, millisecond scale. You're hearing language, and you're able to understand it quickly. Right? So it's kind of cool that happens. Now, the next topic are G protein coupled receptors. And I'm just going to jump into what they look like. So here we have a receptor where a drug is on the outside binding to the drug binding region of the receptor, transmembrane spanning region. In fact, there are 7, and we'll talk about that on Friday. and there's the cytoplasmic domain. Cytoplasmic domain lacks enzymatic activity. And so this drug binds and causes a conformational change, but has no enzymatic activity on its own. Down here, we have an effector enzyme. And so here is an effector enzyme. Here is the drug binding. How do we go from conformational change to action on effector enzyme? We use a coupling molecule, something that couples the conformational change to the effector enzyme. And so that coupling molecule is called a G protein. And so I've drawn it here to look like the letter G. But in fact, it doesn't really look like the letter G. And it associates with the cytoplasmic domain, so that when the drug binds, it senses the conformational change. And it picks up a molecule of GTP, which it uses as energy to move away and to act as an effector. So the reason why it's called G protein is just like a common name for GTP binding protein. And the GTP is the energy source for the movement of the protein. So there are many types of G protein coupled receptors. It's estimated that there's at least 1,000 different ones or subtypes in humans. These are some representative ligands that use G protein coupled receptors, and I'll point out catecholamines here because we talk about catecholamines a lot in later portions of the course, but also things like glucagon, histamine, follicle-stimulating hormone. They're going to act by activating the G stimulatory G protein. GS stimulates an effector enzyme called adenylyl cyclase, which causes cyclic AMP levels to increase within the cell. And so the way it kind of works is no drug, no effect. Right? The G protein likes to associate with the cytoplasmic domain. It's bound by GDP, but it's inactive. Drug binding causes that G protein up a GTP molecule, which is going to give it energy to move away. It's going to move the effector enzyme. And in this particular drawing, it's stimulating the effector enzyme. So the activated G protein stimulates the effector enzyme. Here the effector enzyme is adenylyl cyclase. Okay. Here the effector enzyme is adenylyl cyclase. So cyclic AMP levels increase. The signal is terminated not by drugs falling off, but by hydrolysis of the GTP. So when the GTP is hydrolyzed, the G protein goes back to the receptor and awaits another signal. Okay, so in this diagram we can see the G protein bound to GDP goes back to the receptor and waits for another induction.
G protein coupled receptors lend themselves to a mechanism that separates ligand excitation of the receptor from G protein mediated activation of the effector. So what does that all mean? Well, if you look here in the upper left, you see that there's a G protein coupled receptor not activated. In the upper left, immediately below that we see agonist binding, the G protein picking up a GTP molecule, and now in the bottom left acting on the effector enzyme. And notice what has happened from the bottom left to the bottom right. The G protein is still acting on the effector enzyme even though the drug has now fallen off. Right? So once the ligand activates the G protein to release its GDP and to bind GTP, it does not matter whether or not the receptor ligand is bound or free. What is the rate limiting step in the termination of the signal is the hydrolysis of the GTP back to GDP. So in theory, if you look at the bottom panel again, the drug can bind the receptor, G protein gets active, the drug can fall off, the G protein is going to stay active until hydrolysis of the GTP. So in the upper panel here, if the hydrolysis of GTP happens quickly, there'll be less effector enzyme activity and less second messenger than if it takes longer to hydrolyze, as you see in the middle panel, or if it takes even longer to hydrolyze, as you see in the lower panel. So the rate-limiting step in the termination of effector activation is the hydrolysis of GTP back to GDP. So this is why a neurotransmitter like norepinephrine may encounter its membrane receptor for a short period of time, as short as a few milliseconds, but its signal may last for tens of seconds. Okay, because yes, it can bind and fall off, but those G proteins that have been stimulated and are acting on effector enzyme will stay stimulated until the GTP is hydrolyzed. Okay, so do you feel comfortable with that? Again, all I'm really trying to tell you is that what dictates when the signal stops after the ligand falls off is the hydrolysis of GTP. The family of G proteins contains several functionally diverse subfamilies, and each subfamily mediates effects of a particular set of receptors and is connected or hooked up with a distinctive group of effectors. So for example, if we look at the G protein, it is going to be coupled with different types of receptors, such as receptors for the beta-adrenergic amines like catecholamines or to glucagon, for example. And these receptors, when stimulated, are going to activate the G protein, which is going to act on the effector enzyme, adenylyl cyclase, to increase the activity or stimulate that enzyme activity, and cyclic AMP levels will increase. Alternatively, the G protein will inhibit adenylyl cyclase if the G protein is stimulated. So the stimulated G protein will inhibit adenylyl cyclase and lead to decreased cyclic AMP. And if it's happening in the heart, as we would see with the muscarinic receptor in the heart, acetylcholine will bind and stimulate the receptor. The GIG protein will get stimulated, but its action on adenylyl cyclase will be to reduce the activity of adenylyl cyclase. Cyclic AMP levels will fall, and in the heart, potassium channels will open, which will lead to a decrease in the heart rate. And so there are several types of receptors that are coupled to GI, among them the alpha-2 receptor and one of the muscarinic receptors, because there are some muscarinic receptors coupled to GQ. And so those two receptors in particular, the alpha-2 and the muscarinic receptor, will be receptors that we discuss again later in the course. Now there are odorants that bind to receptors in the olfactory epithelium, things that smell nice, things that may not smell nice, which mediate the signal through the olfactory G protein, which also stimulates adenylyl cyclase. And there are other G proteins, most of which we won't focus on, but there are other types of G proteins like G0 or GO, as they call it, or GT, for example. Before we forget about G proteins, can't forget about the GQ G protein, which is really, of them all, the big three are GS, GI, and GQ. And that's coming up on the next slide. So the GQ G protein is going to stimulate the effector enzyme phospholipase C, and phospholipase C in turn will upregulate levels of second messengers such as IP3, which is also called inositol triphosphate, DAG, which is also called diacylglycerol, as well as cytoplasmic calcium levels. Okay, and so these are going to result in pharmacologic responses in cells and tissues. Last but not least is the GT G protein, and this is coupled to a special type of receptor which has photons as its agonist. And this G protein coupled receptor that we see in the eye is coupled to GT, which when stimulated will affect the cyclic GMP phosphodiesterases, increase their activity, decrease levels of cyclic GMP second messenger, and play a role in the sense of vision. Again, I'm not going to focus on GT per se, but it's an interesting type of coupled receptor because its agonist is actually not a drug, but a light wave called a photon. So we left off last class talking about G protein. We had just gotten up to this idea of G protein coupled receptor. And today I want to start by telling you a little bit about what this receptor looks like. And to just give you an idea, there's something like 800 G protein coupled receptors in our genome. It's estimated to be about that many. So what's interesting about these 800 genes is that they code for proteins that all kind of look like this to some degree. And so if we were to look at this, we'd see that this is the outside of the cell up here, our cell membrane in the middle, with these Roman numerals kind of in the middle, which would represent transmembrane spanning domains. And then if we look on the inside, in the cytoplasm, we see these loops. We've got 1, we've got 2, we've got 3 intracellular loops, with the 4th intracellular region being a long carboxy tail. And so early scientists who worked on G protein coupled receptors called these things serpentine receptors because they kind of look like a serpent or a snake wiggling through the membrane of the cell. And what we know about them now is that we have these 7 transmembrane spanning regions, and what you're seeing here is a 2-dimensional picture of really a 3-dimensional structure. And the way these arrange is such that the amino terminus is on the outside, right? The drug binding domain is going to be formed as this thing wraps the 3 dimensions here in the extracellular portion. And on the inside, we've got 3 intracellular loops and a tail. And I'm just going to call your attention to the 3rd intracellular loop, because the 3rd intracellular loop is where the G protein associates. Remember how we drew this picture last class that the G protein associates with the receptor? Well, it associates with the receptor at this tail. This loop, which is close to the carboxy tail. So there's going to be something I'm going to talk about now that involves the 3rd intracellular loop, the G protein, which is not pictured here, and the carboxy terminus. Now, like I said, there's something like 800 G protein coupled receptors in the human genome. Some very typical ligands for these receptors include the adrenergic amines like epinephrine, norepinephrine. Acetylcholine works by acting at a type of G protein coupled receptor called a muscarinic receptor. Of course, we learned last time, or recently, that acetylcholine can also act at a ligand-gated ion channel, right? So acetylcholine, one agonist, can have different receptors. That ligand-gated ion channel was called the nicotinic receptor. Right, so like I said last time, you may see ligands on multiple lists. And when that happens, it's because you can have a ligand that has more than one receptor in the body. Right, so acetylcholine, the muscarinic receptors look like this. The G protein coupled receptors in your nose that sense smell look like this, as well as G protein coupled receptors for serotonin in your brain and in your vascular system. Many peptides work through GPCRs, G protein coupled receptors. Opioids work through these types of receptors. Even photons, as I mentioned very briefly previously, can stimulate G-protein coupled receptors in your eye and transfer that photon from a ray, the wave particle, into vision. Right, so this class of protein receptor, the G-protein coupled receptor, is very important in pharmacology. So again, I'll just point out that we have an extracellular amino terminus, or carboxy terminus in the cytoplasm. The receptor looks like a snake going through the membrane, and there's going to be an interaction between the third intracellular loop and the tip. Now, again, I mentioned that the left panel is a two-dimensional picture of a three-dimensional receptor. And what I've tried to do in the right panel here is give you that three dimension. Of course, I can't really do that on a flat slide. But what I've done is I've taken those transmembrane spanning domains with their Roman numerals, and I've kind of made them look like cylinders here. And the only thing that's really missing are the polar head groups and the hydrophobic tails. So this is sitting in the membrane, and it's showing you how it wraps around in a circular way. And the amino terminus is where? On the outside, right? The carboxy terminus is where? On the inside. And so if you look, I've drawn these red portions to represent the intracellular loops, and these white portions to represent the extracellular loops, which are not going to be something we talk about right now, but the intracellular loops are important in the G-protein interaction. So this is kind of what it's looking like in the membrane, if that's helping at all. And so if this dashed line now represents the cell membrane, and this area where the agonist is would be the outside of the cell, and then this area would be the inside of the cell, then what you can see here is that when the receptor is not activated by drug, the G-protein associates with the third intracellular loop. Again, it's not covalent interaction, but there's an association there. And so the drug will bind to the receptor, and the G-protein will pick up a GTP molecule and will have energy to move towards an effector enzyme. Right? So we've talked about that. And so that effector enzyme is going to make second messenger. In this instance, we haven't labeled what the effector enzyme is. It might be adenylyl cyclase, right, making cyclic AMP. It might be phospholipase C, making phosphoinositide, calcium second messengers. It could also be cyclic GMP phosphodiesterase, depending on the G-protein. What is the effector? So the effector is going to be linked to the type of G-protein. So if it's a GS G-protein, what's the effector? Which enzyme? Adenylyl cyclase. If it's a GI protein, which enzyme? Adenylyl cyclase. So the I means inhibitory. Inhibitory of what? Adenylyl cyclase. So here we show the enzyme getting active. So this would probably be GS or GQ. If this was drawn as the G-protein coming over and inhibiting the effector, it would probably be GI, inhibiting adenylyl cyclase. Does that make sense? We've talked about this now a little bit. Right? So while this kind of mechanism is generally conserved from one G-protein coupled receptor to another, what differs is the G-protein that's coupled and the effector that's either activated or stimulated or inhibited in some cases. So now remember last class we said, well, this is going to keep going until the GTP is hydrolyzed. Right? So now the drug has fallen off the receptor. So the inactive G-protein comes back to where? The third intracellular loop. And it waits for the next stimulus. Right? So that's the typical cycle here. Well, I wanted to point out that in 3 dimensions, this carboxy tail, or this cytoplasmic tail of the receptor, actually sits in close proximity to the third intracellular loop of the G protein. And so do you remember how we said that molecules can kind of vibrate or wiggle a little bit because they have a certain energy associated with them? So this tail isn't just fixed in a conformation that's far away from the G protein. It can oscillate a little bit. And then there are certain things that can happen to the tail that make it flip towards this third intracellular loop. And when the tail flips towards the third intracellular loop, it blocks the G protein from accessing that area. So this tail can be interference, right? It's like in football when you have the receiver is going to catch the ball, and then the defender pushes the receiver down, right? Pass interference. So you never have the reception, right? So here, the tail can wiggle towards the third intracellular loop, block the G protein, but that only happens under certain conditions. So, remember last class we talked about something called receptor desensitization, where a receptor can find agonist and at first respond, and then, even though agonist is bound, the cell or tissue stops responding. Right? And I drew that last time as receptors bound to agonists being endocytosed and being put into cytoplasmic vesicles that can either be degraded or recycled to the surface. And so we said desensitization as a phenomenon is a response of the tissue where actually it doesn't respond anymore. So it responded to the drug, it stopped responding to the drug. Right? It becomes desensitized. Right? So it's kind of like if you've got a younger sibling when you were young, right? And they kept coming in the room because they wanted to play with you, but you were trying to study or do your homework. Eventually you got desensitized. You just stopped listening because you had something you had to do. Right? Desensitized. The noise was there, the request was there, but you didn't follow through because you had to do something else. In pharmacology, tissue can be stimulated, but under prolonged stimulation, it might stop responding. And so that's called desensitization, different from downregulation. Remember downregulation, the receptor was brought in, and it went to fusion with the lysosome, and it got degraded, and then the cell had to make a new receptor, and that took a lot of time. Here, desensitization is usually temporally faster and reversible. So usually by a few minutes after stimulation, the tissue can respond again. Okay, so the desensitization is temporally shorter than downregulation would be. And so how do we even know about this? Well, again, going back to the experimental science portion of pharmacology, people started working with tissues and receptors in vitro and measuring responses in vitro. And one of the first types of studies that was carried out on second messengers was carried out with cyclic AMP. In fact, cyclic AMP was the first second messenger ever discovered. It was discovered by someone named Earl Sutherland. And he worked on this second messenger. And what we learned from his studies, other than the fact that second messenger does things in the cell resulting in signaling, is that we also learned that tissues can be desensitized. So how does that look? Well, if you were to look at cellular levels of cyclic AMP on the y-axis of this graph, cellular levels of cyclic AMP over time. So the x-axis here is time. Okay? Different than what we've talked about so far. And if you were to look at this green rectangle at the top, this would be indicative of adding agonist to the culture dish at a concentration that binds the receptors. Right? Adding agonist into the culture dish. And I'm leaving it there for this amount of time on the x-axis. And so do you see what happens? I'm adding an agonist here to the culture dish. And what happens to levels of cyclic AMP very soon after addition? Go up. Is that what you would predict if this was a GS-coupled, G-protein coupled receptor? Yeah, right, you would predict that. And then you would probably think that as long as the agonist is there, the levels would stay high. But what experimental pharmacologists saw was that in the continued presence of agonists, the tissue stopped responding and levels fell back to normal. Even though the agonist was there, even though the agonist was bound to the receptor, the tissue stopped responding. Right? And now we know what happened is that the agonist-bound receptor is internalized and put into a vesicle where it's not able to function anymore. And either it's going to be degraded or it's going to be recycled back to the surface. And so scientists decided to let some time go. Right? So they took the drug out of the tissue bath. Right? They washed out the tissue, added fresh culture medium. Okay, so this is called a passage of a few minutes in the absence of agonist. They washed the tissue, they took out the agonist, and then they added the agonist again. And what happened? The tissue started to respond again. And then it started to not respond again. And so this break in the graph is a few minutes. Right? So the tissue could respond again in a few minutes after first being stimulated, and then continually activated, it stopped responding, it became desensitized. You wash out the drug, you add it back after a few minutes, and the tissue responds again. That's consistent with the model we talked about of recycling the receptors, right? It takes a few minutes to recycle the receptors back to the surface. If you had receptor downregulation and a few minutes go by and you add back the agonist, what would the curve look like? Would it look flat? How many people think it would look flat if you had receptor downregulation? I think it would look flat, right? Because you don't have receptors. They've been degraded. The cell's making new ones. Can the cell make new receptors in a few minutes? No way, Jose, right? It's going to take a few hours. Can the cells shuttle receptors back to the surface if they've just been internalized and then recycled in a few minutes? Yes. Yes, it can. So this process of desensitization is temporally shorter than downregulation, but it's a phenomenon that's been known to happen for certain receptors. And one of the receptors that happens for, that we clearly understand the mechanism, is the beta-adrenergic receptor. There's another receptor that it happens for, the nicotinic receptor, and we don't actually understand the mechanism that clearly. We know it happens, we see it pharmacologically, right? But we don't understand it that clearly. So I'm going to talk to you about desensitization again in a later chapter when we talk about drugs that can paralyze skeletal muscle. Those are called skeletal muscle relaxers. And at that time, I'm going to tell you some of those drugs actually bind the receptor, activate the receptor, but the receptor desensitizes and no longer responds to the drug. Nicotinic receptor. So write that down somewhere. Nicotinic receptor. But for the near future, for the next few minutes, I'm going to talk about the beta-adrenergic receptor. Again, why? Because we clearly understand the mechanism here. Another reason is it's a receptor we talk again about after the midterm when we talk about sympathetic drugs. Drugs that can either stimulate the sympathetic nervous system, sympathomimetic, or drugs that can block sympathetic responses. Things like alpha receptor blockers, beta receptor blockers, and in fact, beta receptor will be a topic that we're going to know a lot about before we move on this summer. Move on to the summer. Okay, so although many kinds of receptors undergo desensitization, usually the mechanism is obscure, like in the case of the nicotinic acetylcholine receptor. But in the case of the beta-adrenergic receptor, the mechanism of desensitization is quite clear. And so what you're going to see in the next slide, I'm going to explain, these words that are here in the next slide as to what's happening.
Now, a few words that you should kind of put in your mind for the moment that you're going to hear in the pathway or the mechanism are cytoplasmic tail, right, which I've shown you this morning. Beta-arrestin, which is a protein that we haven't talked about yet. It's a protein. Beta-arrestin is a protein. It's a soluble protein in the cell. And it's given a name based on what it does. And what it does is arrest the signaling of the beta-adrenergic receptor. So it causes desensitization, or is part of the desensitization pathway. All right, another player we're going to discuss in the picture that's coming is the G protein itself. Another term you're going to hear is cellular phosphatase, an enzyme that cleaves phosphate groups. That's also present in the cell. We haven't really talked about yet. And there's going to be another protein in this mechanism called BARC. And we haven't talked about that protein either. BARC stands for beta-adrenergic receptor kinase. Beta-adrenergic receptor kinase. So, how does the beta-adrenergic receptor exhibit desensitization? What is the mechanism? Well, on this slide, now I've put some of the other names on here. Okay, these are proteins floating around inside the cytoplasm, like beta-arrestin or BARC kinase, beta-adrenergic receptor kinase. Now, if it's a kinase, it's going to do what to a substrate? Add a phosphate group. So if this is called beta-adrenergic receptor kinase, what target is being phosphorylated? The beta-adrenergic receptor. And so what's happening? Well, in this model, as you can see, drug has bound to the beta-adrenergic receptor, G protein coupled receptor. The G protein is over here already, stimulating its effector enzyme. In the case of the beta-adrenergic receptor, this is GS. Right? The effector enzyme is adenylyl cyclase. And so what second messenger is going up, up, up? Cyclic AMP. Very good. Right? So the G protein is doing its thing, and that's stuff we've kind of talked about already. But what we haven't talked about already is some other things that can happen. And so as the drug binds and the G protein gets stimulated, so too does a kinase inside the cell. I drew it here as a red thing, meaning it's off. And in response to drug activation of receptor, it turns on. So BARC goes from inactive to active. So it also senses the conformation change. So there's two things happening when the drug binds to the beta receptor. One thing is that the G protein is going over there doing its thing. The other thing that happens is that BARC kinase becomes active. And it phosphorylates the beta-adrenergic receptor. And it phosphorylates it in a very special place. It phosphorylates it on the tail of the receptor, which is rich in serine and threonines. So what kind of kinase is BARC kinase? Is it a tyrosine kinase, or is it a serine threonine kinase? The serine threonine kinase. Right? So BARC kinase, in response to drug activation of receptor, come up to the tail and phosphorylate the serine and threonine residues in the tail. And I've drawn that here as these three yellow phosphates being put onto the tail. And I don't know if you've noticed, but I've drawn the tail as having flipped now. Right? It's exhibited a conformational change. Remember, phosphorylation does something to a substrate. It changes it a little bit. And so the tail wiggles or moves or changes its conformation closer to the third intracellular loop. Right? At this point, that other protein, beta-arrestin, gets involved. So beta-arrestin has an affinity for the phosphorylated tail.
And so beta-arrestin is going to come up to the complex, as so. And it's going to form a beta-arrestin phosphocomplex that's now going to obstruct access of the G protein to the third intracellular loop. Right, so it would be as if you put a big brown bear in front of the door. And the G protein is a person trying to get into the room. And the big brown bear is blocking access to the room. Right? So this beta-arrestin phosphate tail complex is preventing the G protein from coming back. And so again, remember, the G protein stays active until the GTP gets hydrolyzed, right? So in this situation, the GTP got hydrolyzed, drug is still bound, G protein would go back and get reactivated, right? It would go back and it would pick up a GTP molecule and shuttle back to the effector. If it could, but it can't. Why? Because it's blocked, right? So this is why, even though the drug is bound to the receptor, the effect goes down. Right? If there was no desensitization going on, this thing would keep going back and forth. This thing would stay on all the time. You would have a plateau. You wouldn't have a peak and a fall, the graph that we just saw. So this is what we understand to be the mechanism. So as that G protein, it got hydrolyzed, the GTP got hydrolyzed, it's trying to come back to the third intracellular loop. It's blocked by the complex of beta-arrestin and the tail. And so, even though drug stays bound to the receptor, the signal falls. Now, when the drug actually falls off the receptor, this beta-arrestin phosphocomplex can be reversed. And it gets reversed by a type of enzyme, an ACE, called a phosphatase. Now, why do we have to use an enzyme to reverse this? Because phosphorylation is what? What kind of bond is phosphorylation? Is it ionic? Is it hydrophobic? Is it covalent? Right, you need an enzyme to put it on. It takes ATP's last phosphate and shoves it onto the substrate. How is the phosphate bound in ATP? Is it covalent? Yes. Right? So these are covalent bonds. So they just don't fall off. Right? The phosphates just don't disassemble once they've been put on. You need a machine to break them off. Right? And so the machine is called ACE phosphatase. Phosphatase cleaves phosphate groups off of the substrate. And so phosphatases are found inside cells. And they kind of look like guillotines. Right? So if this stringy looking thing is a protein with a phosphate group on it, right? It's almost as if a phosphate is like a guillotine that chops it off. Right? And so as you can see the phosphate in action and the phosphate group being removed from the protein. Of course, in real life, phosphate cleavage doesn't result in blood being released. But I guess I had some time on my hands this day when I made this slide. Right? But anyway, do you understand what phosphate does? It's a cleaver. It cleaves phosphate groups. When the phosphate groups fall off the tail flips away from the third intracellular loop. And when it flips away from the third intracellular loop, what can sneak in and come back to that area? The G protein. And so the receptor is ready to go again. Right? So drug falling off activates phosphatases. The phosphatases cleave the complex, and the G protein can gain access. So we don't need to make a new receptor. We don't need to synthesize. We don't need to spend hours to days. Right? So we can wash out the drug, reverse the process. A few minutes go by, the receptor's ready to go again. That's receptor desensitization. However, if the drug was to just stay there all the time, we wouldn't reverse it. Right? It would stay desensitized until the drug falls off,
and the receptor has a few minutes to reset. Does that make sense? Now, in very prolonged activation of beta receptor, you could actually see receptor degradation. All right, so that is possible. Beta receptors can be degraded as well. They can be internalized and fused with the lysosome. But that would describe receptor downregulation, not receptor desensitization. So the difference is temporal. Desensitization, the tissue can recover in a few minutes. Downregulation, the tissue needs time to make new receptor and insert it. Okay? So hopefully what you're seeing here is that the tissue is not static, right? It's dynamic. It's responding to things. Right? It has a way to respond to things. It has an ability to respond. So, any questions on desensitization per se? Right? Just to review, the mechanism involved, some old information, right? The G protein having gone over to the effector and doing its thing, and some new information, this bark kinase that can also be activated by agonist binding. Phosphorylates the tail, beta-arrestin comes in, and physically obstructs access to the G protein. That's why it can't come back and get reset until the complex falls apart. And that can only happen when the drug falls off, when the receptor is given a break for a few minutes. Okay, so receptor desensitization differs from the slower onset and more prolonged effect of receptor downregulation. And just to remind you, downregulation is an agonist-induced decrease in the number of receptors through internalization and degradation. So it takes time to make new. Any question on the two? Okay, I think that's the last slide of the packet, yes? All right, so this information presented in this course is based on reference materials that are believed reliable and consistent with standards accepted at the time of presentation. Possibility of human error and ongoing research and development in medical sciences does not allow assurance that the information contained herein is in every respect accurate or complete. The information presented during the course is intended for teaching of undergraduate and graduate students in biomedical education and is intended only for teaching. Students who successfully complete this course should check the product information sheet included in the package of any drug they plan to administer or dispense to be certain that the information presented in this course is accurate and that changes have not been made in the recommended dose or in the contraindications for administration. July 9th, 2014.